Hi, everybody. Thanks so much for joining us today. My name is Tom, and I'm an educator here at Cheehaw Park and Zoo, Flint River Aquarium, and Thronatiska Heritage Center. And today we are going to be talking about a topic that is very interesting and cool to me personally, and I hope you all enjoy it and get a little bit inspired as well. So today we are going to talk about citizen science, and we're going to talk about what citizen science is, why it's really important, and what some of the big benefits of it are, and then we're going to talk about some different examples, and especially one that's relevant to Georgia. So let's go ahead and get started and talk about some citizen science. To help us do this, I am going to share my screen with you all really quick. So give me 1 second, please. Okay. So let's talk about what citizen science is to begin with first, because it's a really important topic. And so it's this idea that there's public participation and collaboration that goes on within research. So it's not just scientists conducting the research, it's actually the public getting involved and getting engaged and working side by side with scientists and researchers to help increase how much information can be collected and to increase scientific knowledge in general. So citizen science is really cool because it can happen from multiple different angles. You could have researchers and scientists create these programs, and they kind of try to gather people to help them out with their projects, or it could be the other way around. It could be a group of citizens who have this idea or this desire to research something, and they may go to a group of scientists and say, hey, we think we have this cool idea. Do you think this is something that could be done? And so there's lots of different variation in citizen science projects. They vary in intensity, so it can be something as simple as e-birding. So you can go on to apps and you can document if you see a species, and that can be a really widespread and cool way that anybody can participate in citizen science. Or it can be something much more targeted and something much more designed. And so we're going to talk about a couple of different types of projects that are a little bit more targeted towards certain topics and that are a little bit more intense in that they actually require some training for people to be able to participate. Whereas eBirder is something where people can go on their own and participate, and they don't necessarily need to be trained or anything like that. And so I have here some pictures of some wetlands, and you can see there are some teams in these wetlands. And so this is a citizen science project that I participated in up in Churchill, Manitoba in the subarctic. And so there's actually some pretty cool connections between what I'm going to talk about here with this project and the project that's more relevant towards Georgia. And so what's happening in these wetlands is there are different teams that are collecting lots of information on these wetlands. And so it's a little bit of a long-term monitoring project for these wetlands up in the subarctic to see how they're changing over time within the context of warming. So as temperatures continue to warm in the subarctic and we lose more snow and ice, how are these shallow wetlands that are important habitat for animals, and they're important, chemically as well. So they are trying to monitor these over time. And so what's happening is there's these teams in there, and every team has a different job. But what's cool is that they've had teams, what these are called Earthwatch teams, that are citizen scientists coming up and monitoring these exact same wetlands every year for about 8 years. And they even monitor the same wetlands several times throughout the summer of each year. And so they measure things like the temperature of these wetlands, they measure things like the pH, they even measure physical characteristics. So they will walk the perimeter of the wetland to get an idea of how large the wetland is each year to see if the size of these wetlands are changing. They'll measure the depth, so they always have to try to walk out to the deepest point and measure the depth of these wetlands. So there's several things, including the physical characteristics, right? Like the size of it, the depth of it, some of the chemical characteristics such as the temperature and how much oxygen is found in the water or what the pH is. And then we also monitor animals that are found in these wetlands. So you'll notice that a couple of these people are in the wetlands with nets, and we have some bins on the side. And that's because we go through and we dip net all of these wetlands, and we bring back whatever is collected in the dip net, and we search it for all of the different animals. It can be invertebrates, it can be vertebrates, anything that is caught in these nets gets recorded. And then we can also monitor changes in animal diversity and abundance over time. So we also record, if we get multiple of the same animal, we'll record how many of each animal we catch. So there's lots of good indicators of how healthy a wetland is per se, and that can be anything ranging from the chemical or biological characteristics that we're collecting information on. And so what I'm going to talk about what's called QA/QC a little bit later in the talk, but essentially that's this idea that one of the biggest concerns with citizen science is that everybody needs to be on the same page with how they are collecting the information they're collecting. So there needs to be some consistency in how information is collected, because if we're trying to monitor changes over time, we need to make sure all the information is getting collected in the same way, and hopefully with a similar level of training from each participant. And so we do go out with these teams. One of the best ways that we kind of try to control the quality of the information we're collecting with these teams out in the subarctic is, first, we have several researchers who have done this a little bit more often that can help people out. If they have any questions, they can feel free to ask us. But we also train everybody on all the gear they use and show them the techniques that they're going to do before they go out and actually start doing it themselves. So it's kind of fun to watch the teams progress over time. The first wetland that they do takes them about an hour, and then by the end of the week, they can get through several wetlands in a single day. And so it's just important to recognize that there needs to be some quality control when having citizen science happen. So that's one really cool example, and that's a cool example of a program that has citizen scientists travel all over the world. So this subarctic site is just one site where a citizen science project with this company called Earthwatch is happening. And it's the big benefit up there is they could not monitor as many wetlands as they could with just the staff they have at the science center up in the subarctic. So having these teams go out and help them collect information on these wetlands really helps make this project possible, because we'll have teams collecting information on all of these different topics at the same time. Whereas if researchers from the science center were going to go out and collect this information, they would have to do everything one at a time, and it would take them essentially an entire day just to do one wetland. So having these participants is very, very helpful. And then the other really nice benefit of citizen science isn't that just we can have a lot of volunteers that, again, make research like this possible, but the other thing is that they're volunteers. So oftentimes they are volunteering their time. You do not have to pay them, so it could be a little bit cheaper. And then lastly, this is a great way to increase awareness of different projects like this, so of topics, things like warming maybe impacting wetlands. And so the big hope is that all of these citizen scientists that come help out with this research, we hope they find it really cool. We also provide them with lots of presentations to help them learn about these wetlands and the different methods that they're using back in the classroom as well. And the idea and the hope is that they will also go share everything they learned and talk to other people about the research they were conducting up in the subarctic to get more people excited and hopefully interested, maybe even get some people to come out. We actually know that between the 2 years I was there anyway, a couple people who came the second year were encouraged by people that were on some of the teams the first year. So it's really cool to see how it can have an effect that trickles down further into just the general public, which is the whole idea of citizen science, is that it's everyday people coming to help out if they're interested. Okay, so that's one example. And then really quick, while we're still talking about some of the subarctic work, so I also had some citizen scientists help me with my research. And so again, the big thing that I like to kind of have taken away from this is that a portion of my research would not have been possible without the citizen scientists, because again, it was a type of project that required many people to be involved. And so essentially we were monitoring or studying differences in temperature conditions, but on land more so rather than in the wetlands. And so really we wanted to study these wood frog habitats at multiple locations in the subarctic, but to really make a direct comparison between the different types of habitat, we needed to have people studying the different measurements at the exact same time to really be able to make direct comparisons. So for example, this top left picture is showing tundra habitat up in the subarctic, and so you can see that's defined by much more low-lying vegetation such as moss and lichen, not a whole lot of structure in terms of trees or shrubs. And so then there's also, you can see here at the bottom though, your boreal forest habitat, which is pretty characteristic of what a forest looks like in terms of these taller cone-shaped trees. And so really we wanted to monitor differences between the two different habitat types at the same time. So having those citizen science members who could help use these plaster models to study things like water loss rates of frogs in different habitat types was really important work for understanding how the subarctic may be changing in the future. Okay, so lastly, we have another project that also focuses on the wood frog a little bit. And so, but you can also see that there's two other amphibian species here. So there's also a program that happens in Maine that trains citizen scientists to go out and look for the egg masses of these amphibian species within shallow wetlands called vernal pools. And so this is very helpful because these amphibians rely on these shallow wetlands that are only temporary. So we call them ephemeral wetlands, and that's because they dry up each year. But these amphibians have specialized to lay their eggs in these wetlands because there's not things like fish that are going to be predators. So it's a really safe environment for them to lay their eggs in. But oftentimes these wetlands are overlooked because they are only temporary each year. And so there may be some concern that they may get developed over. And so these three species that rely on these wetlands for laying their egg masses can be used to justify protecting some of these wetlands. And so what can happen is you can go out and do egg mass counts, and if there's enough eggs between these three different species, a wetland can get considered for protection, and so it'd be considered a protected vernal pool. But this can take a long time too to go out and do these egg mass counts and try to get to all these different wetlands and evaluate them. And so people volunteer their time to go out and do an initial assessment of these wetlands, which is really helpful and cool. And so that's very nice because then they can provide information and proof, things like pictures of the eggs, and bring that back to researchers and scientists and say, okay, we found here eggs here. We think this is worth you going to check out and doing a proper assessment of this wetland now. And so what's nice is I just mentioned another type of quality control that can happen in a project like this, which is people can simply take pictures and provide the proof and the evidence that these amphibians are there to really help save researchers and scientists time. So they won't go reassess a wetland unless some pictures are provided to really prove that this wetland is worth checking out. And then it's nice because this can lead directly to the protection of wildlife habitat if we have enough volunteers going out and providing information to biologists to say, you know, these wetlands are out here. So a really cool example of how even citizen scientists can lead to some habitat protection, which is really cool as well, and not necessarily just contributing to some of those longer term monitoring programs, such as in the subarctic. That's really more aimed at understanding some concepts such as how wetlands and wetland characteristics are going to change over time, but rather this one's a little bit even more applied in that it can lead to some direct protection, which is really nice. And it's really cool. It's something that citizen scientists can really get excited about. And so, but this project, there is also training provided. So we train people prior to them going out to their wetland, what the egg masses look like, how to distinguish between the three different egg masses for the three different species, you know, how to properly take pictures while keeping the eggs safe, and all of these different things. And then we train them even just to do some physical observations as well. So what shape is the vernal pool? What's the depth? Can you estimate the size of the vernal pool? What types of plants are there? And all of these different types of characteristics, which is a pretty common theme for monitoring like this, to get some general characteristics while people are out there. Okay, and then lastly, we can move on to an example that is much more relevant for Georgia itself. So this is called Adopt a Stream. This is a statewide citizen science water quality monitoring program. So it's very cool. It's similar, like I mentioned, to the subarctic wetlands in that it is citizen science and it is monitoring over time. So this one is also more long term, but this is very cool because this is a great way to demonstrate how citizen science can really help expand the potential of how much information can be collected and in a much shorter period of time. So the idea that this is statewide and it's done by citizen scientists means many more stream locations can get monitored over time. And so what will happen is after somebody like myself goes through the training to become a, a participant in this program, then I can go out and select a spot, and that will become my spot that I will monitor over time. And so you can go online and you can actually see the map of where people are monitoring currently or in the past. And so you can see the whole extent of where different sites are being monitored within the watershed. And then I provided here what they have as their adopt, and this shows really what their goals are as a program. So it's to do things such as raise awareness, which is what I said was really important for the subarctic too. We are hoping people are going to go back and teach everybody what they learned about in the subarctic, to collect water quality data. So that's a big one too. The whole point is to collect information through this monitoring, to gather general observations as well. So there are physical, chemical, and biological monitoring that can happen within this program, to encourage partnerships and get more people involved and to support this kind of work because it's really important for monitoring water quality within our state. And also one of their biggest goals as a program is to provide the tools and training that's necessary to get citizen science members out there. So this includes things like a PowerPoint day where they teach us what their goals are and why this type of work is important. They train us on how to collect all of the different variables that we do when we go out to our site, and they provide the tools necessary for us to do these collections. And so I have there that there are multiple ways to assess water quality over time. There can be physical monitoring, so you can just make observations about the physical stream and surrounding environment itself, such as how the stream bed looks adjacent to wherever you're monitoring as your direct site, to see if there's things such as erosion taking place over time. You can assess the vegetation surrounding your site. There can be chemical monitoring. So this gives you a snapshot into what the chemical conditions of that water are at the time that you go out each month. And so this can be things, again, such as your DO, which is your dissolved oxygen. This is really important because you can get an idea as to what the oxygen content is in the water. This is important because lots of animals need oxygen for respiration and breathing. So it's a really important chemical condition within your site that you're going to monitor each month. There's pH, so there's temperature, which is also related back to dissolved oxygen. There's conductivity. There's lots of things that you can monitor and measure each time you go out to really get that snapshot. And the biggest portion of this that's really cool to me is that if you go out and you monitor the same spot and you measure the same conditions each month, then you're going to be able to notice if there's any big shifts in the trends of the characteristics and conditions that you're measuring. And so the idea is that each day you go out each month, the goal is to go out at the same time of the day because time of day could impact, conditions such as the temperature. And if you go out at the same location, the same time of day monthly, you'll be able to see trends over time. And then the goal or the idea is that if something were to pop up that seems abnormal or irregular, then you can really start to focus on that spot and figure out if something is going on or if there is potentially something like a point source type of pollution that is now being introduced at this site that has maintained fairly consistent conditions. But then if something pops up and there's a change in something like conductivity that it really is spiking now, then that could be an indication that something has been introduced to the stream area that you are found at. And at that point, you could bring that to somebody else and say, okay, now I've monitored this change, and I think this is worth checking out. And so it's kind of nice what I was talking about with the other project, where the citizen scientist can bring the information to someone at a higher level, to say, I monitored this change, and so I think it's worth assessing further and getting a second set of eyes to really evaluate this. And then there's also biological monitoring. So this is really nice too. You can, I kind of talked about this is subarctic wetlands, that we could do the dip netting and get an idea of what the invertebrate community was looking like in these wetlands, and monitoring changes in the abundance or the diversity of animals within these stream sites. And so changing diversity of animals within a stream site can be a really good indication of whether or not you still have a healthy stream. So some animals, like invertebrates, are pretty sensitive to changes in water quality. And so if you notice an animal group that's been there for months or years and it starts to disappear, that's a really good indication that something has changed at your site, and can be a third way of kind of providing some monitoring. Okay. So I've kind of mentioned this, that the idea is to regularly collect data at the same location at the same time. And then lastly, another type of monitoring that we can do at these stream sites, which is really cool, is we can collect water samples and bring them back to evaluate them for E. coli. So this is a really good way to determine if there is any type of really bad contamination at our site. And so just to give you a kind of an idea of one process that we go through when we are doing this water monitoring, is we will take baggies out with us, we will collect a water sample, and we will also collect what we call blank. And I'll talk about why that's really important, because that comes back to that quality assurance, quality control that I was mentioning earlier. And what we'll do is we'll bring our water samples back in a very controlled environment. So we'll bring them back in a shaded cooler. And then within just 24 hours, we have to get our samples running. And so what we'll do is we'll come back and we'll do what we call plating. So we'll take just 1 milliliter of our sample, and we'll put 1 milliliter on each plate. Or so this is a petri dish. This isn't one we actually use, but this gives you a really good idea as to what it looks like. And then we have to do what's called, we have to incubate it. We have to warm them. So we kind of have to like bake them at a very specific temperature for a very specific amount of time. So it is all controlled and consistent each month with your sample, right? And so after you have your 3 different samples, so we will do 3 different plates, all from the same water that's collected, to kind of have some replication as well, to make sure that there's nothing too wild happening in one versus the other. And then we'll actually average what we count in each of those 3 plates. But then there will also be a 4th plate, and what we'll do is we'll plate our control, or our blank is what I called it earlier. And so what that does is that just makes us confident that we didn't have any contamination happen during this process. So our blank should come back without anything in it, to help us be confident that the way that we collected our water samples wasn't contaminated to begin with. And so what will happen is, after they have cooked essentially for that set amount of time at the very specific temperature of 35 degrees Celsius, what we can do is we can go through and we can count these different dots essentially. So what we'll have in our samples is some red dots and some blue dots. And so what we're looking for is any of these blue dots that have what looks like an air bubble surrounding it too. And so that's really important, because that indicates that the bacteria is still alive and giving off carbon dioxide. So we won't count something unless it is blue and it has that entrapped gas next to it to indicate that it is actually a live count. And so what we'll do is we'll go through and we'll actually count how many we have, and then we can calculate how many we have what we call CFU, which is colony forming units, within our sample for 1 milliliter. And then we'll average, like I said, the 3 different plates, and we can times it by 100 to get an estimate of how many colony forming units, which is what we call it, of E. coli are found within a 100 milliliter sample. which is kind of our standard for monitoring E. coli concentrations. And so this is a good way to monitor changes in contamination to ensure that we don't have any contamination from things like animal waste or a leak or anything like that that may be introducing contamination into the water. And then essentially we'll monitor this over time. And if something goes over 1,000 CFU per 100 milliliters, that's when it needs to get brought to the attention of somebody, because this is now a contamination concentration that's high enough to be concerned about, because E. coli can make people sick if they're in the water. And so at that level, if we monitor over 1,000 CFU, that's when we'll bring it to the attention of someone else, and that site will really start to go out and get assessed to see if they can identify the source of the pollution or the contaminant and where it's being introduced and how it can be addressed to help get that water quality back to a cleaner and safer type of water column. So this is one really cool way of how we can do monitoring through this program over time. And then, like I mentioned, people can go in, they can get trained to do this as part of the quality assurance, quality control. You have to actually do a test to see if you can get counts that are pretty representative of what you should be finding. And that's one way that they can really say, okay, this person picked up what they learned during their training and they are able to apply what they learned, and now we are confident that they can go out and help do some water quality monitoring, be part of this team. And then, like I said, anybody can go pick a site and they can regularly go out monthly and start to collect this information. And then that's just one more site that's helping to be assessed so we can get a bigger picture of water quality throughout Georgia. And then hopefully, people will go out, they will tell other people, they will increase the awareness, maybe get other people excited to join and really spread this program and make it more effective. So I hope you all took away from this some of the really cool benefits of citizen science, how it can really help expand scientific knowledge just by having the general public out and participating. And I hope you all learned something and enjoyed this talk. And thanks so much for joining us today. Take care, everybody.